Good morning, children. Today we are going to study about the cilia and the flagella. Both are locomotory organs and they are different from each other. They are different from each other in many respects, but their internal structure is similar. Okay, morphologically different. But similar internal structure. Now, what is the difference between the two? This we are going to discuss now. So for that, I'll write here cilia and I'll write here flagella. So the difference between the two. Now, if we consider the example where these are present. So the example, if we consider this is present in paramecium and this is present in euglena. Yesterday, I made the structures for you. I think you still remember what I made, how I showed you in the structurally the euglena and flagella. Do you remember or do I have to make it again for you? Do you remember it or do I have to make it again for you? Yes, beta, you remember? Yesterday I made it in case of euglena and paramecium. You still remember that? Okay. Okay. Rohan and Tadar has answered. Rest. 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 Do you remember? Yashika, okay. Deepika, okay. Fine. So this is, they are present here. Now, if we see their size, if we see their, first of all, location, I'll tell you location, where these are located or the site. These are present all over the body, paramecium all over the body. And euglena, in the flagella, in case of euglena, it is present on the anterior end of the body. Now, anterior end like this, I made euglena for you like this, and this is the anterior end, this is the posterior end. So the flagella was present here at the anterior end of the body. This is what it means. Now, another point of difference is their number. So their number, when we consider their number, the cilia are many in number, or they are numerous. Whereas if we consider flagella, they can be 1 to 3 flagellas are present, or 1 to 4. Okay, flagellas, these are present. Now, if we consider that their size, what is their size? They are very small. The cilia are very small, and their size is about 10 to 15 or 5 to 10. Write down 5. Yesterday I told you 5 to 10 micrometers. So this is their size, and they are larger than cilia. And their size is about, say, 150 micrometers. Their size is about 150 micrometers, and they are larger than cilia. Now, the next part which I told you is how do they beat, the beating of the locomotory organs, or how do they move. We'll write beating of locomotory organs. So beating of cilia here and beating of flagella here. I'll write here the beating of flagella. So when we see this, it shows two types of movement. I told you it shows two types of movement. One is the first one which I told you is synchronous type, synchronous, and the second one was metachronous type. Synchronous is when the longitudinal flagellas move. This is movement of or beating of transverse flagellas. Sorry, longitudinal flagellas. Longitudinal, not flagella, cilia. Longitudinal cilia, they're beating, and beating of transverse cilia is metachronous. Whereas in case of this, they beat independently. The flagella, the beating is they beat independently. Because they are few in number, located at the anterior end, so they beat independently. There is no such synchronous or metachronous type of movement seen in case of flagella. The next point. The next point of difference is how many types of cilia are present and how many types of flagella are present. So we have done location, we have done number, we have done size, we have done beating. Now we are on to the movement. If we see the movement in the case of this, it is they show the cilia show rowing movements. Rowing movement. Rowing movements means it consists of two strokes. The first stroke is, like in swimming. The rowing movement consists of two strokes. One is power stroke, another is recovery stroke. By the power stroke, what is done is that the water is pushed back. Like in swimming, what we do when we swim, with one stroke, we push the water and then we take it forward and then again recover and then again we push the water. That means when a swimmer is swimming, when suppose this is a swimmer who is swimming, first of all, it takes its hands on the either side and cuts the water. Okay, and then from the top, it brings its hands back and again push the water back. So this is what the power stroke and the recovery stroke. That means they bring their hand like this, push the water, bring their hand from the top again, push the water. This is recovery stroke, and when they push the water, that is power stroke. So two types of strokes are there: power stroke and recovery stroke. So these two types of strokes are seen when the cilia function. In case of the flagella, we see undulating movement, or it is also known as wavy movement. Wavy movement. Or undulating movement. Like this. If this is flagella attached here, so it will show a movement like this. That means a wavy movement. If it is attached here, it will show a wavy movement. Like this. A wavy movement or undulating movement is seen in case of flagella. So this is about the movement. Now, if we see types of flagella, types of cilia and types of flagella. So the next point I'm writing here is types. There are two types. These are also two types, and the flagella are also two types. Which two types are Cilia and which two types of flagella? The two types of cilia are kinocilia and stereocilia. Kinocilia, I'm writing here K-I-N-O, kinocilia and stereocilia. These are the two types of cilia. Kinocilia are motile. These are motile, that means they show movement, whereas stereocilia are non-motile. They do not show movement. Then what is their function? What is the function? This I'm going to tell you. The kinocilia, these are, motile. That means I'm making the example of stereocilia here. Like they are present in paramecium. Suppose this is paramecium. This is paramecium. The cilia are present all over the body, and a number of cilia are present at the cytopyge or the gullet or the mouth. A number of cilia are present. Now what happened? These cilia which are present here, they show the movement, special movement, and because of that special movement, the food from here is taken in. Food from here, from outside, is taken in, and this is able to feed itself. So the motile cilia, they help in bringing the food, they help. And also, if these are these longitudinal ones and the transverse ones, when they move, they help in the movement of the, because of the recovery stroke and the power stroke, they help in the forward movement of the organism. So these motile cilia, which are kinocilia, they help in the movement and the capturing of food, because these are motile. The another type of cilia, which is stereocilia, these are non-motile. Then what do they help in? These stereocilia, they are attached to sensory organs. Sensory organs. Example, taste buds. They are attached to taste buds. Now what happens when they are attached to taste bud? If suppose this is the taste bud, they are attached here at the top, the cilia is present, and from another end a nerve fiber is present. So whatever the perception is, it is received by this, and then further from the nerve fiber it goes to the brain. So this is what they help in, the sensory observation, the sensory feelings rather, we say. So they help in various senses, conduction of various senses wherever they are attached. Whereas kinocilia, they help in movement. One more example of this I'll tell you of kinocilia is fallopian tube or oviduct. Fallopian tube. Another is trachea. So fallopian tube and trachea. On the inner wall of fallopian tube and on the inner wall of trachea, the cilia are present. And the cilia, in case of fallopian tube, they help in the movement of the egg or the zygote. See, the egg is present in the fallopian tube. So how it is brought from the ovary? See, it is captured by the fallopian tube when it is released from the ovary, and then inside it moves to a junction where the sperm comes and fertilizes it. So how it moves to that junction? It moves to that junction because of the kinocilia. So the kinocilia, they are present in fallopian tube, which helps in the movement of the egg. And after the egg is fertilized, the zygote formed, it goes and attaches to the wall of uterus. So how that fertilized egg, which is zygote, it moves from the fallopian tube to the uterus. It moves by the help of kinocilia. And trachea on the inner wall of trachea, some cilia are present, and those cilia, they capture the dust particles, the pollens, so that they may not enter our lungs. So this is the function of this. And this, they are attached to the sensory organs, so they help in conduction of various impulses. Is it clear the types of cilia, or any doubt or any query in this, the types of cilia, beta? Yes. Yes. Okay, Priya is saying it's clear. To others also, is it clear? Fine. Now the types of flagella. I told you these are also two types. The two types of flagella are, number one, whiplash types, and number two is tinsel type. Whiplash type of flagella is simply like this. little broader from the base. Like this, it's like a hunter. And it helps in movement. Whereas the tinsel type is similar to this plush type. The only difference is it has a number of bristles on it. These are bristles which are present on it. And these bristles, they help in further locomotion of this. See, all the organisms where the flagella are present, in 90% the flagella are whiplash type. Say Euglena, in the Euglena also whiplash type of flagella is present. This tinsel type of flagella, these are present in brown algae. In brown algae, and which particular structure? Zoospores. The zoospores or brown algae, they have this tinsel type of flagella. So is it clear the difference between cilia and flagella, beta? Anything which you could not understand or any query in this, you can go through it once, just what I have written on the board, and any query or anything which you could not understand, you can ask me. Yes, bache? Anything in this which you are unable to understand, you can ask me, or any question, go through it. I give you 2 minutes to go through it. I give you 2 minutes to go through it. Yes, beta. Then the last topic, this is over. After this, I'm going to start with biomolecules. Yes, one thing I would like to tell you, today is 10th and from 12th we have our, I think, holidays. For 5 days will be closed. So what I want to say that today this chapter is over. So tomorrow, can we have a seminar type of session of this chapter? Anybody who would like to present any topic tomorrow? Yes, who will present a topic? You prepare one topic and present it in a symposium way. Yes, who's going to do that? Yes, beta? Who's going to do that? No volunteers. I'll have to speak the name. Yashika did last time, I remember. Amrita has also done last time. So right now, and Priya has also done last time. Am I right, Priya? You have done it? Yes, you have done it, na? Last time when I asked you to prepare a topic, Yashika, Amrita, and Priya have done. So this time it can be Rohan, Deepika, Tadarani, three of you. I think we'll have only, or one more. I'll speak the name. You can prepare one topic each, and I'll give to every child about 10 minutes. So Deepika, Rohan, Tadar, and one more. Anybody who wants to do it on its own. Jasleen, is that okay? Jasleen, will you be able to do? You can prepare any topic and you will have to speak for 7 to 10 minutes of this chapter. Yes, beta? Okay, Deepika is saying she will prepare nucleus. Yes, she has given the topic also, Deepika. Any other? Deepika has given the topic also. Rohan, what topic have you selected? Yes, Rohan? All the topics are easy, am I right? I'll go with cell organelles. No, but all the cell organelles, how can you explain? All the cell organelles need detail. Mitochondria itself will see. Today also I'm doing one of the organelle and the whole time goes in one organelle. So I'm saying that I'll give you, okay. Rohan is saying endoplasmic reticulum. That's fine. Rohan will be preparing endoplasmic reticulum. Now, Jasleen, your topic. Jasleen, what will you prepare with? Okay, ma'am, I'll prepare. I'll tell you on WhatsApp. Fine, fine. You prepare yourself. And one more name I gave, Tadar. Tadar? Tadar? Cilia and flagella. Fine. She is saying cilia and flagella. Fine. So, Bete, you will prepare 7 to 10 minutes topic. We'll do recording and this will be put on the whatever we record, that will be put on the group tomorrow. Okay. So just prepare it very well. Fine. Okay. So, beta, this is what the difference between the cilia and flagella. I'll tell you the internal structure of this very quickly, which is not much difficult. If you see, the internal structure is given in the book and it shows a cartwheel shape. If you see a big circle, it is somewhat like this. Yes. Yeah. So inside, this is given in the book. If I draw it like this, if I join this, it comes to be like this. It comes to be like this. And in this, if you see, there are, in the center, we have a structure like this, two semicircles rather, one here, one on another side. Let me show a little more in circular fashion. Like this. Like this. And then we have, in the center, we have two circular structures, one at like this, another on this side, which is like this. And both the circular structures, both the structures, these are joined by a bridge. Like this, a bridge joins the two structures. Then on the periphery of this, we have various microtubules. One is towards the inner side, which is complete. This is complete circle. It should be a little bigger. One is like this. You can see the complete circle. And another is half circle. Like this. Complete circle and half circle. And this way we have 1, 2, 3, 4, 5, 6, 7, 8, and 9. We have 9 of such. If you have your book, you will find out that we have 9 of such. These structures which are present here, this structure, this circular structure, and this circular structure, both these circular structures, these are known as microtubules. These are known as microtubules. The microtubules which are present in the center, are known as central microtubules. Central microtubules. I'm just writing this. And the ones which are present on the periphery are known as peripheral microtubules. So we have two types of microtubules, central microtubules and peripheral microtubules. In the center, we have a bridge which connects these two microtubules, and this bridge is known as interdoublet bridge. This is inter-doublet bridge. This is inter-doublet bridge. Now, if you see the microscopic structure of these microtubules, it appears to be like this, one complete and another half, and they have their diameter is, they are like this. If you see the internal structure, they are like this. They have space here, and in this space are present, in the complete one, which is this complete circle, in this there are 13 protofilaments. I have made 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. There are 13 protofilaments. These are known as protofilaments. And in the second, which is half visible, in this we have 10 protofilaments. 2, 3, 4, 5, 6, 7, 8, 9, and 10. Here we have 10 protofilaments. Now, the microtubule which is present in the central microtubule is of this type. That means it has 13 protofilaments. So, if we look at it, it will appear to be like this, which is having 13 protofilaments inside. It will have 13 protofilaments inside. Now, if we see this the complete one, this one which is complete, and this one which is complete. This is made up of tubulin protein, made up of tubulin protein. And this one, the one which is half, this is made up of dynein protein. So the two proteins which are present in them is tubular protein and the dynein protein. So these are just a minute, beta. I have done this. This is dynein protein and this is tubulin protein. Just, I'm sorry. This is dynein protein, D-Y-N-I-N, dynein protein, and this is tubular protein. The half one is tubulin protein. Tubulin protein and dynein protein, they are made up of. Now, the pattern in this, the pattern of cilia and flagella. If you see, we have two microtubules in the center. Okay, 1 and 2, and we have 9 around. Now, that means the pattern which we see is 9 plus 2. We have a pattern which is 9 plus 2 pattern, which is seen in cilia and flagella, and 9 microtubules which are arranged around, which are peripheral, and 2 microtubules are central. Now, bache, now this structure which encloses these two, the structure which encloses these two central microtubules, this is known as central sheath. So this is central sheath. And we have various, if you have your NCERT book, you will find here from the peripheral microtubule, there are some spoke-like structures which come out from each microtubule. And these, if we make one more here, if we make one more microtubule here, from here also one spoke-like structure will go out. These spoke-like structures are known as radial spokes. Radial spoke. So these are the spoke-like structures which are known as radial spoke. And we have this, which is plasma membrane. This is plasma membrane. And this complete, the outer membrane is plasma membrane, and the structure which is inside it, this is known as axoneme. Axoneme. The structure which is inside the plasma membrane is known as axoneme. Now what happens? When this is a cartwheel type of structure, when this cilia or flagella, it has to move. It has to move. In case of cars, if you see, if this is the tire of the car, if this is the tire of the car, it will move only when the central thing will move. This central structure, when it moves, then only the complete tire moves. So same thing happens here. For the movement of this complete structure, this has to move. So first this moves, and then mm, the complete this cartwheel type of structure moves and helps in the locomotion. Now how will it move? When it has to move, these radial spokes which arise from these peripheral microtubules, they attach the central sheath. They get attached to the central sheath. They